Hello, and welcome to this video on describing protein structures. At the end of this video, you'll be able to describe the 4 levels of protein structure in chemical terms, explain how secondary structures protect hydrophilic atoms in hydrophobic spaces, and recognize several ways to present protein structures. So proteins are described by 4 levels of structure. And these 4 levels of structure are called primary, secondary, tertiary, and quaternary, but they are not hierarchical. In fact, as we'll see, the secondary and tertiary structures in particular are highly interconnected and interrelated based on their chemistry and the timing of which these kinds of folding occur. To start, all proteins are made of amino acids connected together by covalent bonds. And the primary structure of the protein is just that linear string of amino acids. So we have seen before that when you have 2 amino acids, and in this picture you can see you've got the N alpha carbon C double bond OOH, and the same on the amino acid that's next to it. We can do a condensation reaction between the hydroxyl group here and one of the hydrogens of the next amino acid, and that will form a peptide bond here between those two. And if we continue doing that, we could get a long string of amino acids, one after another after another. On the right here, you see a string that goes histidine, aspartic acid, leucine, tyrosine. And this linear structure of amino acids is called the primary structure. Here's another presentation of this. I just wrote my own name, DANAHL, in amino acid. As you look through, you can see all the way it goes N, C, C double bond O, N, C, C double bond O. So if we were looking at this, that means that this side chain is D, aspartic acid. This one up here is A for alanine. And then you've got N, another alanine, histidine over here, and leucine all the way out here. So there are 20 amino acids that get put together in all kinds of combinations, and that linear string that you can read is the primary structure. We know, though, that proteins don't really exist in the cell in that linear primary structure. Instead, they fold to create bonds that stabilize the structure in the aqueous environment of the cell. Because proteins exist in water, the hydrophobic amino acids need to be secluded. And we've seen this before. In this picture, you can see all of the non-covalent interactions that are happening here between these full charges. This is an ionic interaction between 2 side chains. Here's some hydrophobic interactions happening between all of these methyl groups here as you zoom in. And then you see a hydrogen bond over here between these 2 amino acids. So the folding of a protein, the 3-dimensional structure, depends on non-covalent interactions between the side chains of that protein. And this is called the tertiary structure. So the interactions that happen between side chains and other side chains, or between the side chains of the amino acid and the backbone, are called the tertiary structure. And just to remind you again, when you look at an amino acid, N, C, C double bond OH, there's always a hydrogen here. And then there's a part that hangs out over here, this R group. This is the side chain. So the chemistry that's possible in this tertiary structure depends on the chemistry of that side chain. And remember, some of them are hydrophobic, some of them are polar and uncharged, some are positively charged, and some are negatively charged. The overall composition of those within a protein will dictate what kinds of folding are possible, what kinds of tertiary structures will exist. But what happens to the hydrophilic backbone atoms that are in that hydrophobic region? Those backbone atoms are still connected to the side. For example, if we had several amino acids linked together, N, C, C double bond O, N, C, C double bond O. We'll do one more, N, C, C double bond O, and we'll put our OH group out here. And let's say that there were side chains on all of these alpha carbons, and they're all hydrophobic. So this part of the protein with those hydrophobic R side chains hanging out on the outside might fold into a conformation where all that hydrophobic stuff gets tucked way into the inside of the protein, away from all the water on the outside. But even if it does that, it has to take with it all of these atoms here in the backbone. Those nitrogens, all of these oxygens, and those are very polar. You can't just tuck all of those polar atoms into a hydrophobic space because they themselves are not hydrophobic. So what do we do about that? In a membrane protein, this will become super important because all of this space right here is very hydrophobic. If I wanted to put a protein through that membrane, I would have to do something about all of these polar nitrogens and oxygens that make up the backbone of my protein. The way we solve this is by having those backbone atoms interact with each other in a couple of common ways that shield those hydrophilic atoms in hydrophobic spaces. The one that's shown here is an alpha helix. And so what you see as you go around this helix, the nitrogens are shown in blue. So this is NC C double bond O, NC C double bond O, NC C double bond O. And what you see is that the nitrogens and the oxygens are aligned as we go around this helix, NC C double bond O. Here's another alignment. Here's another alignment. And so by doing that, we create hydrogen bonds between the nitrogens and the oxygens that are in the inside of this helix. And because they're in the inside of this helix, all of the R groups can point out. You see that a little bit better in this diagram, though it's a little bit crowded. So again, as we go around, NC C double bond O, NC C double bond O, you see this oxygen here in red is having a hydrogen bond with a nitrogen further down. And so everywhere you see these dashed lines, there's a hydrogen bond happening between the nitrogen of one part of the backbone and an oxygen of another part of the backbone. And all of the side chains you see in purple are sticking out away from the helix where they can interact with a lot of hydrophobic stuff. So if we go back one picture, now we see that this green protein in the middle of the membrane has all of its hydrophilic atoms down the middle of this helix and all of its hydrophobic side chains sticking out, interacting favorably with the hydrophobic part of the membrane. The other way we can do this is with what's called a beta sheet. Beta sheets also involve hydrogen bonds between backbone atoms, between oxygens on the carboxylic acid and nitrogens of the amine. But instead of arranging them vertically, they're arranged kind of horizontally in these pleated sheets. So when you think about the word pleats, you might be thinking of something like corrugated sheet metal. You might be thinking about the pleats you would put in a skirt. The point is that it kind of goes up and down and up and down. And in doing that, we align, again, the nitrogens with the oxygens so that they can create these hydrogen bonds and stick the side chains, in this case, up and down and up and down in ways that they can interact with hydrophobic stuff in the protein or in a membrane. So again, the secondary structure requires interactions between those backbone nitrogens and oxygens as a way of shielding and protecting hydrophilic atoms in hydrophobic spaces. And we often present this with a ribbon model where the helices are these ribbons over here, these kind of twisty ribbons, and the arrows represent the beta sheets. So that takes us through the ways in which an individual protein can be structured. It's got its long linear chain of amino acids that folds up such that the side chains can interact in tertiary structures and the backbone atoms can interact in secondary structures. But sometimes more than one primary structure will interact together, and we call that a quaternary structure. So in these pictures, you see two or more individual primary structures. In this picture, you see one which is shown in green and one which is shown in brown. And they're coming together and they're interacting non-covalently kind of along this plane here where these alpha helices are all interacting. And when they're together, they act as a single unit. They were transcribed separately, they were translated separately, they were folded separately, but then they come together into these quaternary structures and function as a single unit. On the right, you see a tetramer where each of these rainbow structures, kind of from red going throughout to light blue, is an individual primary structure that comes together to form the neuraminidase protein. So we can look at these structures in a couple of different ways depending on which information is most helpful. Sometimes it is helpful to see just the backbone atoms so we can kind of see where the protein starts. In this picture, it starts over here. Here's the N terminus. And we could follow this ribbon backbone diagram through red, orange, yellow, green, blue, and ultimately dark blue. And the C terminus of this protein is over here. Sometimes it's helpful to see the secondary structures, in which case we would use a ribbon model, like we saw earlier. And sometimes it's really helpful to see where the atoms actually are in 3D and where there are openings and active sites of enzymes and where everything is really closed up tight. And then we'd use a space filling model. So this is really showing the tertiary structure by showing us where each side chain is and how those side chains might be in position to interact non-covalently and hold the three-dimensional structure of this protein together. Ultimately, we're going to look at a lot of cartoons of space fill models that will help us see the relationships between structures and functions. The shape of these proteins has consequences for their functions, and we'll see lots of examples of that through the semester.